If you were to put a tea bag into a hot cup of water and not stir the water or dunk the bag in and out of the water, what you would notice is that slowly over time that the color of brown from the tea would slowly move throughout the entire cup of water, even though you didn't stir it or dunk the tea bag in and out. Well, how does this occur? Well, movement that does not require any energy is called passive transport, and that is what is occurring in the tea. Many molecules move across plasma membranes passively without using energy, similar to this example. And this type of movement relies on a process called diffusion. This is the spontaneous movement of substances from regions where there is more concentration of solute to an area where there is less concentration of solute. So our solute in this particular example is the T molecules, which move from an area of high concentration close to the bag out to areas of low concentration throughout the rest of the cup of tea. Diffusion always takes place whenever a concentration gradient exists and then continues until particles are distributed evenly. And this even distribution of particles is referred to as an equilibrium. This does not mean that the particles stop moving. In fact, they continue to move randomly at equal rates in all directions, so that they remain evenly distributed and at equilibrium. So in our final cup of tea here, these particles are not staying still. They're constantly moving randomly in all directions so that the tea remains a consistent colour. Lipids and small non-polar molecules such as oxygen and carbon dioxide diffuse easily across the hydrophobic portion of a biological membrane. And cells maintain concentration gradients for these molecules by continually consuming these substances as they diffuse into the cell, or by producing more of the substances so that they then diffuse out of the cell. So these substances are moving with a concentration gradient. So for example, mitochondria consume oxygen as soon as it diffuses into the cell through the process of cellular respiration. This maintains the oxygen gradient that drives the diffusion of oxygen into the mitochondria. Respiration produces carbon dioxide, so you have a high concentration of carbon dioxide inside the mitochondria, which then diffuses out of the mitochondria because a concentration always remains higher inside the cell than outside. Charged particles such as sodium and chloride ions and relatively large molecules such as glucose and amino acids cannot pass freely through the hydrophobic part of the phospholipid bilayer. Instead, they transport across the cell membrane through transport proteins that help the solutes cross. And this process is called facilitated diffusion because the proteins facilitate or assist the movement of these molecules across the membrane. Facilitated diffusion is still a form of passive transport that does not require energy because substances are being moved from an area of higher concentration to an area of low concentration down a concentration gradient. So on the left here, you have an illustration of a cell membrane. And on one side of that cell membrane, you have a high concentration of blue spheres, which represent a type of solute. And on the other side, you have a low concentration of this particular solute. And this solute is able to move freely through the phospholipid bilayer, which means it's going to move from the area of high concentration to the area of low concentration down a concentration gradient. And the only difference between that and facilitated diffusion is that facilitated diffusion occurs through transport proteins, as shown on this diagram on the right. There are two types of proteins that are involved in facilitated diffusion, and these are carrier proteins and channel proteins. Carrier proteins bind to specific molecules on one side of the membrane, change shape, and then release the substances on the other side of the membrane. So that's illustrated here. You can see that certain substances that are unable to pass directly through the phospholipid bilayer approach and bind to a specific carrier protein. That carrier protein then changes shape and transports the molecule inside the cell. So an example is the glucose transporter protein, which is located in the membrane of all mammalian cell types. And glucose is a sugar that is too hydrophilic to pass freely across the phospholipid bilayer. Instead, the glucose transporter protein carries glucose in either direction across the cell membrane, depending on the concentration gradient. And without this, we would not be able to get the essential energy we need to survive. Channel proteins, on the other hand, form narrow passageways through which small ions can diffuse rapidly from a high ion concentration to a lower ion concentration, and only ions of a specific size and shape can pass through particular protein channels.